Video 3 of Unit 1.1 of your IB Biology Study Guide. In this video, we will still be focusing on our introduction to cells, but now we'll be looking at the evolution of multicellular organisms on a cellular basis, and we'll focus on differentiation in cells and also stem cells. So we'll be completing the final points there in your understanding. We'll be looking at the final point in the nature of science. We'll be looking at the final 2 points there in your application and skills. Okay, so in a previous video, we've already discussed unicellular and multicellular organisms. So those unicellular organisms just being single-celled, compared to our multicellular organisms, which are made up of many cells. Okay, so the evolution of multicellular organisms. Now, it's currently believed that life on Earth probably evolved from small single-cell organisms. Now, this picture behind here is of what we call some stromatolites. And it's believed that in these kind of coral structures, found in only several places on the globe, one of which is in Western Australia, small stromatolites lived. Now, stromatolites are single-celled organisms, and they are believed to be the first organisms on Earth. So it's believed that from those unicellular organisms, all life evolved. Now, over the course of evolution, some of these cells have then clumped together over long periods of times and began to work together, developing simple multicellular organisms. Then it's believed that the following evolutionary steps probably occurred. The organisms grew because they no longer were limited by the size of the cell, so that surface area to volume ratio that we discussed in an earlier video. The cells in such organisms were then able to specialize through differentiation, so adapt for their different roles. Multicellular organisms then displayed emergent properties, and we'll talk more about emergent properties shortly. But basically what it means is that the organism can do more things than the individual cell alone is capable of. So here we've just got an infographic showing an idea of the believed evolution of cells, so starting with the unicellular protocysts there, moving on to where they've joined together as a colony, and then it's more an organized colony as such. You see you've got the back cells there, which are the movement cells, the locomotor cells, and the front cells here, they're actually the food synthesizing cells. So you've got this kind of independent unit that operates alone but is a group of cells. And then from there, later organisms have started producing gametes from that so that they could then reproduce. So if we have a focus on this idea of these emergent properties, okay, so the characteristic of the whole organism, including the fact that it's alive, is known as an emergent property. And these emergent properties arise from the interaction of the component parts of a complex structure, basically saying that the whole is greater than the sum of its parts. So if you had an individual cell, it can do less than if you put all the cells together in the multicellular organism. Okay, and this saying that pots are fashioned from clay, but it's the hollow that makes the pot work. But you can't have the hollow unless you have the clay. Does that make sense? Good analogy there. So organisms that carry out temperature homeostasis by using hair or feathers, sweating, changing their blood flow, can exploit more habitats than ones that cannot change their temperature. Okay, so by having different factors, you can get different outcomes. Okay, so let's take this one step further now, because if we're saying that these multicellular organisms with cells that perform different roles are more adaptable as such, how do we get the cells to be able to perform different roles? And that's what we call differentiation in cells. Okay, so specialized cells can develop from multicellular organisms by a process called cell differentiation. This means to become different and therefore specialize in a particular function that could be different to another cell. By becoming specialized, the cells of a tissue can carry out their role more effectively than if they had many different cells. So basically what we're looking at here is a stem cell which can then differentiate into whichever type of cell the body requires. Okay, and each individual cell is more useful in its role than the original stem cell. Okay, so the genetics behind that. Every cell in your body contains the exact same DNA, whether that's your body, whether that's a sheep's body, whether that's a tree out in the yard. Each cell of that organism is identical in terms of DNA. This DNA has the instructions for your entire body, okay, and that's what we call your genome. So every cell has the full set of instructions for your entire body. That's the genome. Each cell has all the chromosomes and all the genes. Now, there are approximately 25,000 genes in the human genome. Every cell has every single one of them. However, not every cell is always expressing every gene. So although every gene's there, depending on the role of the cell, only certain genes are expressed. When a gene is being used in a cell, we say it is being expressed. So that term, expressed. Most of the genes will be switched off, okay, as they will code for functions that that cell doesn't do. All right, so for example, liver cells will have the genes for eye colour switched off. The gene's still there, but it's not being used. And when you look at this diagram here, it's showing that only the genes for liver cells are switched on. Everything else is off. The genes that are effectively switched on are those associated with keeping the cell alive. So for example, respiration, producing enzymes required for survival, and those associated with the specialized function of that cell. So now let's focus a bit more on this concept of the stem cells. Okay, a stem cell can become any one of the 220 different cells in the human body. And just bearing in mind, you get stem cells in all organisms, okay, or most organisms, but we're specifically looking at the human body. Okay, so the stem cell, a definition. An undifferentiated cell of a multicellular organism that can form more cells of the same type indefinitely, and from which certain other kinds of cells arise by differentiation. Okay, so the capacity of stem cells to divide and differentiate along different pathways is necessary in embryonic development. It makes stem cells suitable for therapeutic uses. So basically what this is saying is, for embryos to develop, stem cells are vital because it depends what functions are required in the embryo development for which cells need to be made at which times. However, if those stem cells can be extracted and used to develop cells, specialized cells, it's making a whole new area of research and science, and holds a lot of potential in terms of disease resistance and fighting of certain diseases. Stem cells can divide again and again to produce new cells. They're useful for the growth of tissue and replacement of cells that have been lost or damaged due to whatever factors. They have the ability to differentiate along different pathways to produce different types of cells. They are really, really multifunctional. Okay, so what are the sources of a stem cell? We've got our multipotent stem cells here. Okay, now these guys are also known as your adult stem cells, and they can be taken from bone marrow. So you may have heard of people donating bone marrow for stem cells. These will differentiate into different types of blood cell. Blood in the umbilical cord will also differentiate into any type of blood cell. So our multipotent cells are these that will differentiate into our blood cells. These stem cells allow for limited use in particular tissues. Then we have our pluripotent stem cells. Now these are also known as the embryonic stem cells, and they're taken from a blastocyst or zygote. Okay, so they are taken from the young developing embryo. They can be differentiated into almost any different cell type, and they can be used in therapeutic applications to treat disease or other health problems. At the top here you'll see the totipotent stem cells. Okay, and these have unlimited capability, but it's quite a new area of science at the moment. Okay, so we're just focusing on our multipotent and our pluripotent. Multipotent being those taken from the bone marrow or from the umbilical cord and generally used to make the blood cells, and then your pluripotent coming from the blastocyst or zygote, and these are the ones that can make most different types of cells. Again, just another infographic there illustrating that. Okay, so looking now at some of the therapeutic use of stem cells, and we're going to look at Stargardt's disease, and this is one of your key application tasks. Okay, so the therapeutic use of stem cells, Stargardt's disease. Now, this disease is a genetic disorder and it affects children between 6 and 12 years of age. And it's due to a recessive mutation of a gene called ABCA4. So that is the gene, okay, and it's a recessive mutation. The mutation causes a membrane protein used for active transport into the retina cells of the eyes to malfunction. So we will look further at what active transport means. However, basically a protein that takes certain substances into the retina cells has a malfunction. So the certain substances can't enter the retina cells then. The photoreceptor cells in the retina that detect light then degenerate, and this leads to a progressive blindness. So researchers developed methods for making embryonic stem cells, in turning them into retina cells. So this research has started to be done. So basically what they do is they isolate the cell, they get the stem cell from the human, okay, they transform it into a pigment, and then they can do a transplant back into the eye. So the trials on mice were successful. In 2010, cells were then transplanted into a woman where they attached to the retina and remained there for the length of the 4-month trial. Sight improved with no harmful side effects. More trials are now required. Okay, so our second therapeutic use of stem cells we'll look at is looking at leukemia. Okay, so you had Stargardt's disease and one other named condition. So our one other named condition we'll look at is leukemia. Leukemia is a cancer of white blood cells, or the leukocytes, and it's caused by a mutation of a gene that controls cell division. Leukocytes are made in the bone marrow from multipotent adult stem cells. The leukocytes are released into the bloodstream and basically they fight off infection. Leukemia is when these cells are produced in an abnormally large number. Now, chemotherapy is used to kill the bone marrow cells, the normal cells, as well as the abnormal ones. The bone marrow therefore can no longer produce blood cells. Donor marrow containing stem cells is introduced into the patient's blood. The stem cells then migrate to the bone marrow and start to produce healthy leukocytes. That's the aim. Okay, now what are the ethical considerations we need to think about of using stem cells? So this is your ethical consideration. And here we have some opinions supporting stem cell research against some opinions against stem cell research. Okay, so at the end of that video, we've now covered all of our points that we needed to in 1.1. Obviously still with a little bit of work to do in class.